Okay, it's ready.
Greetings, everybody. Welcome, Bob. Uh, I guess last time we had a really quick intro, so this time I'll just let you know that Bob and I worked together at Gallo. I can't even remember how many years ago. It was probably like 2001, 2002 when we first
Yeah,
met. And uh, I think the best part about Bob, besides all his knowledge, is that he helped me finish writing my master's thesis. So thank you for that,
that's right.
Bob.
It's
He
not
read
about
it. He read every chapter as I wrote it, and he's the reason I graduated.
yeah.
Anyway, thank you for that, Bob. Uh,
Yes.
why don't you go ahead and introduce yourself, and then anytime anyone has any questions, you're welcome to raise your hand or put it in the chat, whatever works for you. We can ask as he's going along. There's no need to wait till the end. So
Yes,
thanks, Bob.
of course. Yes, please ask any questions during the seminar. Yeah, so I'm Bob Tracy. As I mentioned before, I worked in the wine industry for about 25 years. I have a small wine laboratory that's part of my business, and the other part of my business is actually conducting sanitation audits at wineries. So I have quite a bit of experience auditing wineries, all the way from the crush equipment to cellar equipment, bottling equipment, drains, floors, walls, water, atmosphere. So I know where stuff, the wine spoilage microbes tend to lurk in the wineries. So what I want to do today is to kind of the last seminar was all about wine microbiology, to kind of introduce you to all the microbes, make sure we were all up to date on what they do and how they can spoil product. The next two parts today and next time I do one, it's all about winery sanitation. And so I'm going to provide some background, and then we'll talk about the risks associated with sanitation, and then the benefits associated with sanitation, and kind of provide my recommendations. So today I've split the sanitation into two parts. And today what I'd like to do is talk about giving you an overview of winery sanitation, some definitions associated with winery sanitation, the major factors influencing the effectiveness of cleaners and sanitizers. So what helps and what hurts cleaners and sanitizers as far as effectiveness. The types of cleaners and sanitizers used in the wine industry, the recommended properties for cleaners and sanitizers. So what types of things make cleaners really effective and what types of things make sanitizers effective. I'm going to be talking about sanitary design principles for winery equipment and infrastructure. This kind of gets overlooked a lot of the time, like how the design of the equipment is so that you can effectively clean it. So I'll be talking about those principles and how important they are. How to monitor the effectiveness of sanitation. Visually, can you look and see if something is visually clean or dirty? We're going to talk about ATP testing, and we're going to talk about microbial swabbing. And then I'm going to talk about the common sanitation risks that I have identified in wineries. So when I do audits, these are the things that I see in wineries where I know there's risk points in the wineries. And so I'll be going over those risk points with you. And then the final part of today's seminar is I'll actually provide you with some case studies from my audits showing a correlation between ineffective sanitation practices and poor wine quality. So the winery doesn't do very effective sanitation practices, and it's reflected in the quality of their wine, meaning they have a lot of wine spoilage microbes and they're producing a lot of off colors, off aromas, off flavors. So that's today what we're going to be going through. The next seminar in July, I'll be going over the benefits of sanitation and the recommendations for sanitation. So those are the two topics I'll mainly cover next time. Okay, so let's talk about some definitions here. Cleaning versus sanitizing versus sterilization. Cleaning is the active removal of both inorganic, so like mineral compounds, and organic substance from product contact and non-contact surfaces. So this is the actual removal of the debris from the surface. Sanitizing or sanitization is the practice of enhancing product quality and shelf life by suppressing microbial growth to an acceptable level. So we're not sterilizing, we're just getting microbial growth down to an acceptable level for the wine industry. It's also the practice of preventing contamination and spoilage of product. And then if you're using chemical sanitizers, they're required to reduce microbial populations on a clean surface by at least 5 logs. So if you're using chlorine dioxide, if you're using peracetic acid, if you're using iodine, ozone, those things, they're required to reduce microbial populations by 99.999%. Finally, we have sterilization, which is the complete elimination or destruction of all forms of microbial life, including spore-forming bacteria. We do not need to do sterilization in the wine industry. It's not necessary. First of all, it's very costly to do that. And second of all, like I said last time in the wine microbiology seminar, there's no human pathogens that can grow in wine. So we don't need to sterilize our environment to make sure we're eliminating all microbes. So what we're focused on is cleaning and sanitizing. So what I consider cleaning and sanitizing in the wine industry is a 4-step process. First, you need to do an initial water rinse, usually warm water, to remove all the loose soil and debris. You then do have a cleaning step where you're removing dirt, debris, stains, organic material, and biofilms. And this is either done manually by scrubbing or mechanically by spraying. And then you use the cleaners to penetrate, dislodge, and carry away dirt and debris. So for me, the cleaning step is the most important step in the sanitation process. If you don't clean effectively, the sanitizer is not going to work as effectively. So it's very important that you clean the surfaces before you put the sanitizer on. After the cleaning step, you have a rinse step where you're removing residual debris and cleaning chemicals. And then you can sanitize. So once the equipment is clean, the sanitizer is used to kill the microorganisms that are remaining. It's important that sanitizers by themselves do not effectively remove dirt, debris, and soils. So if the equipment doesn't look visually clean, do not go ahead and sanitize it. You need to re-clean it. The sanitizer is only effective on the surface of the stainless steel, for instance. It's only if you have organic debris there, it'll sanitize the top of the organic debris, but it won't get down to the stainless steel and sanitize that. So that's why I'm saying cleaning is the most important part of sanitation. So what are the major factors influencing the effectiveness of cleaners and sanitizers? Time. Increased cleaner contact time, almost always you're going to have a greater amount of soil removed. Increased sanitizer contact time, greater microbe killing power. That's almost always the case. Increase your time and you're going to have more effective cleaners and more effective sanitizers. Unfortunately, in many industries, in the food and beverage industries, time is critical, and so they need to keep moving. So you need to find the best cleaners and the best sanitizers that will help you reduce the amount of time. Temperature. Typically, increased temperature leads to improved cleaning power. So if your temperature is higher, like hot water, steam, your cleaning is going to be more effective. Chemical concentrations. It's important to remember that chemical concentrations vary for the cleaners and sanitizers. It depends on the chemical used, the type of organic soil, and the equipment to be cleaned. And also, very importantly, using a concentrated form of a cleaner or sanitizer is not necessarily better. A good example of this, does anybody know what the most effective concentration of ethanol is or alcohol is when you're using it as a sanitizer? There's a certain percentage that's the most effective.
I'll guess 20%.
Okay, it's a good guess. So why wouldn't you use 100%? So let's say, I want to sanitize a valve on a tank and I want to spray it with alcohol. Why wouldn't I just use 100%?
Is it a surfactant issue?
No, that's a good point though, but no. The reason that you do not use 100% ethanol or alcohol is because alcohol actually denates the proteins on the surface of the cell very quickly. And so the alcohol cannot penetrate into the cell to kill the cell. So you're actually preventing the actual killing power of the alcohol by using 100%. The ideal concentration of alcohol is 70% because it has a little bit of water in it. And then that alcohol can then penetrate into the cell and it's not denaturing the cell wall like the 100% would be. So that's a good example of why you don't always want to use the concentrated form of a cleaner or sanitizer. In addition to chemical concentrations, mechanical force. This could be simple hand scrubbing with a brush to actual mechanical scrubbing.
Hey, Lulu, get away from there.
Mechanical force aids in the soil removal and typically reduces time, temperature, and concentration requirements. So if you're actually able to mechanically scrub a surface or just with your hand scrub a surface, it's much more effective in cleaning than just spraying a cleaner on and hoping that it works. So what are the recommended properties for winery cleaners? So usually you have a certain type of cleaner like a caustic cleaner or sodium percarbonate, proxycarb. The most effective cleaners are ones that are called built cleaners. Ones that have these extra properties associated with them. So when you buy a cleaner, you want to get a built cleaner, not just straight sodium hydroxide or potassium hydroxide or sodium percarbonate. And the properties you want associated with the cleaners is, first of all, water softening. Water softeners help remove mineral ions. You want wetting agents associated with the cleaner. This lowers the surface tension of water, so it spreads out easier on a surface. You want chelating agents. The chelating agents bind metal ions to make them more soluble. So there's certain metal ions, to get rid of them, you add these particular chemical agents to the cleaner. Dissolving ability. The cleaner should be soluble when you add it to water if you're using a dry form. Usually increasing the temperature helps the solubility. The cleaner should have surfactants, and these allow a close contact between the soil and the detergent solution. So the cleaner is actually really close to the soil and can actually do its job. Rinsing ability. You should be able to easily rinse away the cleaner after it's been doing its job of cleaning the surface. So all of these properties you really need to have associated with your cleaner, not just straight sodium hydroxide or straight sodium percarbonate. They should have these properties. So when you get your cleaner from a vendor, you should ask them, does it have these properties associated with it? These are the types of cleaners that are used in the winemaking process in the industry. You have acid cleaners like phosphoric acid. This is generally not used in the wine industry as a cleaner because wine is acidic, so it doesn't really help you clean surfaces as effectively. There's certain times when you might want a phosphoric acid used, but generally you don't. There's strong alkali and caustic cleaners like sodium hydroxide and potassium hydroxide. Many wineries are going to potassium hydroxide because it's putting less sodium out into the environment. There's mild alkali cleaners like sodium carbonate, sodium percarbonate, trisodium phosphate. Again, sodium percarbonate is commonly used in the wine industry. Sodium carbonate and trisodium phosphate are used not very frequently. So the most common cleaners used in the wine industry are sodium hydroxide, potassium hydroxide, and sodium percarbonate. And again, you want these to have the properties that I was just talking about. There's also high pressure spray cleaning. We're using a pressure washer. This can be problematic in that if you're pressure washing an area, you're spraying microbes all over the place, getting in the atmosphere and then landing on equipment and landing at other places in the facility. There's also foam or gel cleaners, where you spray a foam or gel onto a surface, let it sit there for a little while, and then you either spray it off or you actually scrub the surface if necessary. I recommend foam cleaners for like harvest equipment. It works quite well, or for floors and drains in your facility. Okay, we just talked about cleaners. Now, what about sanitizers? So these are the recommended properties for sanitizers. First of all, it's important, you want to make sure the chemical sanitizer has been registered with the EPA. It should have an EPA number on it. You need to protect chemical sanitizers from heat and light. So you need to have them in a cool, dark area when you're storing them at the winery. You want to avoid long storage times for chemical sanitizers because they do slowly degrade over time and the concentration gets less. You want to verify the concentration of chemical sanitizers. There's little test strips like pH strips, which you can use to actually determine the concentration of a chemical sanitizer like peracetic acid or chlorine dioxide. So they'll tell you what the concentration of the chemical sanitizer is that you're using. You want to verify the temperature on equipment when using hot water or steam. The sanitizers should have a broad spectrum of antimicrobial action. They should be able to kill yeast, bacteria, molds. An important one, which I'll actually talk about a little bit more detail later, is the chemical sanitizer should break down and inhibit biofilms. There's two sanitizers that are commonly used now in the wine industry that are very good at breaking down and inhibiting biofilms, and that's peracetic acid and chlorine dioxide. The chemical sanitizer should be effective over a wide pH range, so pH 2 to 10, they should be effective in that full range. They should be effective over a wide temperature range. They shouldn't require a post-rinse, so like peracetic acid and chlorine dioxide, for instance, you don't need to do a post-rinse after you apply it, unless you're going to have your wine go directly back into that tank, for instance. Then you would, of course, do a post-rinse. But if you can leave it, let it do its job for a long period of time, the better. They should be environmentally friendly. And a lot of the cleaners and sanitizers now are pretty environmentally friendly. So what are the types of sanitizers used for the winemaking process? Chlorine. We're not supposed to use chlorine in the wine industry anymore, though, right? So does anybody know why?
PCA.
PCA, yes. It's a very effective sanitizer, but unfortunately, one of the byproducts of using it is the formation of trichloroanisole, or TCA. So I strongly recommend not using chlorine in a winery. And if you do have to use it for like treating well water or something, there's filters you can use to remove it before it gets into the winery. Chlorine dioxide. This sanitizer is actually pretty widely used now, especially at the larger wineries in the Central Valley. This is not chlorine. It's a completely different chemistry. And so it has a chlorine attached to two oxygens, and it's a very different chemical compound, and it doesn't break down into free chlorine. But it definitely is a very good sanitizer. Iodophores like iodine, they used to use more commonly in the wine industry, not as much as anymore, and mainly because they kind of stain a surface that brown color. Ozone, it's a very effective sanitizer. For this one, you want to make sure your equipment is very clean, though, because ozone reacts very quickly. And if you have any organic debris there, it'll break down very quickly and won't be able to actually sanitize. So when I use ozone, make sure that surface is very clean. Peroxyacetic acid or peracetic acid, PAA, probably the most common sanitizer used in the wine industry now. Chlorine used to be before the whole TCA scare happened, but now peracetic acid is probably the most common sanitizer used in the wine industry. It's a very effective sanitizer. It and chlorine dioxide, as I said before, are very effective at reducing biofilms on surfaces. Quaternary ammonium compounds, also called quats. This is a sanitizer that I recommend using on floors and drains and walls. It's very effective. It actually, when you apply it and let it dry, it has like an invisible film, and it continues to act for a long time after you apply it. So it's very effective for floors and drains, which is probably the dirtiest areas in the winery. Sulfur acidulated sulfur dioxide. Some wineries still use this as a sanitizer. It's pretty effective, but you have to make sure your pH is exact and the sulfur dioxide concentration is exact for it to be fully effective. Citric acid. This is not a sanitizer. It's a neutralizer. So a lot of wineries will use a cleaner, then they'll do a rinse, then a citric acid, and then they'll do a rinse, and then they'll do a sanitizer. You can actually skip the citric acid step if you're using peracetic acid, because they're both acids. Peracetic acid is a stronger acid, and it is a sanitizer. Citric acid is just neutralizing the whatever cleaner you're using. Hot water or steam, very effective at killing microbes. And then there's ultraviolet light, which some wineries are starting to use to sanitize the interior of their tanks. It is a very effective sanitizer. I've actually done a few different trials with wineries on it, and it's very effective. The issue, like similar to ozone, is the surface has to be very clean, because ultraviolet light does not penetrate organic debris. So that surface has to be clean so it can actually shine onto the stainless steel surface. So a little bit about hot water and steam. The standard in the food and beverage industries is to use steam or hot water. The temperature has to be at least 180 degrees Fahrenheit for 20 minutes to heat and activate microorganisms on equipment. So that's the minimum, 20 minutes at 180 degrees. Now, if you change the temperature, you actually will vary the time that you actually have to use to inactivate the microbes. So if you're doing it at 194, you only need 10 minutes to heat and activate the microbes. If you're at 167 degrees Fahrenheit, you actually need 45 minutes to actually heat and activate. And then if you're down near 160, you're getting up to 2 hours where you have to actually apply the heat before you get heat and activation. So it's very important for hot water and steam that you're actually achieving a temperature for a certain period of time to heat and activate the microbes. Any questions so far? Okay. Oh, I gotta stop for 1 second, sorry.
No problem. He'll be right back.
Sorry about that. Still got a lab to do, work to do.
Hey, Bob, this is Larry Dino. I do have a quick question.
Sure.
You mentioned on the cleaning that if you use proxy carb, I think you had said that you should have like citric acid in it to make it more effective.
No, not for the cleaners.
Oh, okay.
The only reason the citric acid is, so after you've done your cleaning step, is a lot of wineries will use citric acid to neutralize the cleaner like sodium, like proxy carb.
Oh, okay. But you weren't recommending that it's got to be in the proxy carb.
No.
Okay. Okay.
Yeah.
Cool.
Yeah. Good question though, because that I can understand why maybe you thought that. Okay. Any other questions before I talk about sanitary design? Okay. Sanitary design, like I said, when I was going through the agenda, is a very important piece of sanitation that gets overlooked. And so I'd like to go over some of the sanitary design principles and why they're important. So sanitary design is the application of design techniques that allow the timely and effective cleaning of equipment and infrastructure services. So you're more effective, but if you have good sanitary design for your equipment and infrastructure services, you will reduce the amount of time you have to spend cleaning and sanitizing, and the cleaner and sanitizers will be more effective. Good sanitary design improves product quality and operational efficiency in the following ways. It reduces the risk to product quality by preventing microbial contamination and pest ingress. It reduces the time required for sanitation. It reduces the cost for chemicals, equipment maintenance, and equipment repair. It reduces energy and water usage. And it improves productivity due to reduced rework. So if you have good sanitary design for your equipment infrastructure, there's a lot of benefits as outlined in this list here. So first sanitary design principle, equipment and infrastructure should be constructed so that all surfaces are accessible for inspection, maintenance, and cleaning. So you should be able to easily clean the equipment and infrastructure. You should be able to maintain it, and you should be able to clean it effectively. It's very important. If you have cracks and crevices in your equipment, it makes it much more difficult to clean and inspect. Second principle, product contact surface should be made with materials that are corrosion resistant, non-toxic, and non-absorbent. The corrosion resistance and the non-absorbent are very important. You don't want the surface of your equipment to be corroding, because that allows areas where microbes can get into the equipment on the surface. And you do not want to be non-absorbent. So you want the surface to be not be absorbent. It's very important. Third design principle, there should be no accumulation or pooling of liquids in or on equipment or on infrastructure surfaces. So your piping should be free draining. Shouldn't have any 90-degree angles on your piping. Should be able to freely flow. You should have well draining floors. So the floor should be sloped to the drains, no dead spots, cracks, and it should be non-porous. The ventilation systems should be self-draining. So like your air handling systems, those should be self-draining. You shouldn't have liquid accumulating inside the air handlers. The fourth sanitary design principle, you want to eliminate hollow areas to prevent accumulation of pests and microorganisms. So the floor drain should have a cover. You should properly seal doors and windows. Should have screens on the windows. And you want to seal gaps around piping that's entering the buildings. The next one, the equipment should be free of niches such as cracks, pits, gaps, corrosion, open seams, and dead legs. Joints and wells should be flush and free of pits and cracks. So the picture on the bottom there, that's showing you the inside of two different pipes. One that has nice smooth welds. The one on the left has smooth welds. The one on the right, you can see it's kind of pitted and it has some cracks in it. Those are perfect locations for microbes to accumulate, and they'll sit in there. So you want to make sure that not just the outside of the piping, but the inside of the piping has really smooth welds. Also, you want to avoid using threaded fittings. Threads are another area where microbes can accumulate. For ventilation systems like air handling systems, you want to ensure that airflow will be going from cleaner to less clean areas. And the air should be filtered to control the contaminants. So the handling system should be able to filter out at least some of the debris and microbes in the atmosphere. You should design and install utility systems to prevent introduction of spoilage microbes. So your water system should be protected, your air should be protected, and your process gases like nitrogen. So when I do audits at wineries, these three areas I always look at. Is the water system clean? Is the compressed air clean? Are the process gases clean? So I'll actually swab inside of the compressed air line. I'll swab inside of the nitrogen lines, and you would be surprised how many microbes are inside of those. Especially for nitrogen, you expect, oh, there's no way any microbes are growing in there. But yes, microbes can start accumulating in all of these areas. And water is very important because you don't want to be spraying, if your water has microbes in it, you don't want to be spraying those onto your equipment, and then you're introducing microbes onto your equipment, potentially some wine spoilage microbes, and then just regular environmental microbes. If you are spraying microbes on the surfaces, your cleaners and sanitizers won't be as effective. You're reducing the efficacy of the cleaners and sanitizers. So water, air, and process gases need to be thought about when you're as part of sanitary design. Okay, that was sanitary design. So let's talk about how to monitor the effectiveness of sanitation practices. So you want to be able to tell, is my equipment clean and sanitized or not? And there are really three main ways in the wine industry right now that you can do this. You have sensory inspection, which, visual, smell, touch. And this provides an overall picture of sanitation effectiveness. You have ATP, or adenosine triphosphate test, the real-time method to measure the level of cleanliness on surfaces. And finally, you have aseptic swabbing and culturing of microbes, which measures the sanitary state of surfaces. So when I do audits at wineries, this is the technique I used. I actually use aseptic swabbing and culturing, where I'll swab a surface and then go back to my lab and culture the microbes. A very important method that wineries can use is the ATP test, because that'll give you a good indication how clean your surface is, and it'll give you an answer quickly. The aseptic swabbing and culturing of microbes is a good method too, but it takes 7 days for me to grow up all the microbes on the plates. So with ATP, you can actually right away tell, oh, is that surface clean or dirty? So let's talk a little about sensory inspection. Sanitation is monitored by using 4 of the 5 senses. Visually, does it look clean? Smell, does it smell clean? Touch, does it feel clean? Is it smooth? Or taste, would you want to taste that surface? So using all of these together, you kind of get an idea of how clean your surface is. And I'm sure most of you, when you see a surface, and you can tell if it's clean or dirty, or you can smell if it's clean or dirty. So this is a very effective tool to see how clean and sanitized your equipment is, and your floors and drains are. The second method is adenosine triphosphate, the ATP test, which I was just talking about. And this is a real-time method to measure the level of cleanliness on a surface. So what is ATP? Stands for adenosine triphosphate. It's a chemical found in all animal, plant, microbial cells, and product residues. In your cells, it's the chemical used as form of energy. So it's found in all living organisms, including the yeast and bacteria and molds that impact the wine industry. So how does the ATP test work? It's a light test, and it measures the cleanliness of surfaces. An ATP is measured using an enzymatic reaction, which occurs in the tail of fireflies to produce light. So light emitted is proportional to the amount of ATP present. So when you swab a surface with an ATP swab, you swab it and then put it into a little vial. And at the bottom of the vial is the enzymes which are in the tail of fireflies. And when ATP gets in contact with these enzymes, the enzymes will metabolize that ATP and produce light. So it's a very neat little tool to see how clean your surface is. So because if you have ATP on your surface, this test will tell you if your surface is clean or dirty. And the measurement for ATP is its relative light units. That's the measurement tool, measurement test. So several wineries are starting to use ATP, especially on their bottling lines. And I don't know, I thought somebody mentioned that maybe all the Livermore wineries can get together and get a few different ATP test units, and then they can kind of use it
Well,
among.
it's definitely on our mind. So
Oh, it is, okay.
yeah, what I'm thinking is it might be something we can do from the Wine Quality Alliance and then make that available as far as the unit to read the swabs, but it might be easier to have the swabs purchased by the wineries themselves. I'm not really sure how much they cost, but that seems like the best way to go about it, but we can always talk about it more.
Yeah, no, and I think it would be really helpful for the wineries in Livermore to use something like this. And yeah, so it would be good if you had a couple units and then the wineries buy the swabs. That would be, I think, that good way to do it.
Can you answer Alex's question?
Mhm.
What RLU do you consider to be sanitary?
That's a good question because it depends on what ATP unit you're using. So if you guys end up purchasing a particular unit, then I'd be able to tell you what the relative light units specs are for that particular unit.
Alec, do you guys have a meter already?
Yeah, I'll call the lab team and I'll follow up with the unit. Thank you.
Oh yeah, sure. Yeah, because like the Hygienia unit is you want to be less for like bottling equipment, you want to be less than 10 RLUs. And for like cellar equipment, I think it's like 30 RLUs. But then the charm
That's the one we're using.
Oh, Hygienia, yeah. So for bottling, you want to be less than 10 RLUs, and for cellar equipment, it's usually less than 30. And for harvest equipment, it's usually less than 50 RLUs.
Perfect. Thank you, Bob.
Yep, sure. But the one thing to mention is that like the charm unit has much higher threshold for RLUs. So that's why I'm saying the unit's very important. Okay, so we just talked about ATP. Then there's the aseptic swabbing and culturing of microbes, which is the technique I use when I'm doing sanitation audits, because I need to know what microbes are there and how many are there, and so that's why I use this when I actually am conducting audits. So it's a surface-based swab recovery method used to detect microbial populations in winery equipment and infrastructure. It provides an indication of the effectiveness of the sanitization practices. And how does this microbial swabbing test work? So the sterile swab is applied to a 4-inch by 4-inch square area. I transfer the swab to sterile liquid, saline solution, shake it. And then when I get back to the lab, the liquid is filtered through a 0.45 micron filter. And then I put that filter on a Petri plate and the microbes are cultured. And so let's say I'm doing an audit at a winery and I have to collect 100 samples, swab samples. I'm doing this process for all 100 swab samples. And then the hardest part and the most difficult, challenging part is actually reading the plates after they're cultured, because you have a lot of different microbes on there and you have to be able to determine which ones are which and how many there are. So what do swabbing results mean if spoilage microbes are detected? That means your sanitation process is compromised. So if I'm seeing a lot of microbes, wine spoilage microbes on a rack valves of tanks, I know that they haven't properly cleaned and sanitized the rack valves. And one important thing to keep in mind is the ATP results do not directly correlate with microbial counts. So I think people get confused by this. They say, oh, well, I have a high ATP, then I should have high microbial counts. Not necessarily. And so there is no direct correlation between the ATP results and the microbial counts, because you could have a high RLU reading and low microbe counts, or low RLU reading and high microbe counts. Because the ATP readings can be a combination of benign wine residue and microbes. So there could be ATP left over that's not necessarily that was from microbes, but it's still kind of lingering around. Also, ATP can vary widely between microbes. So some microbes can produce large amounts of ATP and some produce less amounts of ATP. And how I always considered it, an effective sanitation monitoring program typically incorporates visual inspection program, ATP testing, and microbial swabbing. So you kind of use all three to tell you how clean and sanitized your equipment is. Okay, so what are the common sanitation risks that I encounter in wineries? So let's talk about effects of poor sanitation. So the presence of fruit, must, juice, skins, seeds, wine residue, and or biofilms can serve as sites for accumulation and growth of microorganisms. So as you're leaving that stuff on your equipment, on the floor, in the drains, you're just asking to have for microbes to accumulate and grow on those surfaces. The growth of microorganisms can lead to deterioration of spoilage wine products. And this is what I talked about in the first session on wine microbiology. Yeast growth can spoil product during fermentation and in the cellar. You can get the formation of acetic acid, ethyl acetate, acetaldehyde. Yeast growth in bottle can cause pre-fermentation. Brettanomyces growth can spoil wines by producing 4-ethylphenol and 4-ethylglycol. You can get bacterial growth causing wines to spoil because of the production of volatile acidity. And then mold growth can theoretically lead to the formation of haloaerosols like TCA and TBA. So there's several, as I talked about in the last session, there's several microbes that can actually spoil product. And if you're allowing them to accumulate on your equipment or on the floor or in the drains, then potentially they could contact your product and then start growing in your product. The other thing to keep in mind is there's microbe cross-contamination. So ourselves, our hands can cross-contaminate. Microbes can get up in the atmosphere from drains and floors, and then land on your equipment, land in your wine. So cross-contamination is something that really needs to be thought about. If you have a surface with a lot of microbes on it, like a floor area or a drain, those microbes eventually will get up in the air and could land on your equipment and in your wine. So biofilms. Biofilms are probably the hardest thing to deal with, and if you get them, it's very difficult. You can actually get pretty good contamination in your product. So just because something visually looks clean doesn't mean it's clean or sanitized, because biofilms can be invisible to the naked eye. What biofilms are, they're a mixed group of microorganisms in which cells stick to each other and adhere to a surface. So what the picture below will show you is the microbes attached to a surface. They start growing, and then they start dispersing off of that surface. And the cells that adhere to the surface are frequently embedded in a extracellular coating of polysaccharide. So that's usually the way biofilms start, is the microbes attach to polysaccharides, and then they start growing on a surface, and they have a nice film on them. And then it's very difficult to clean them. So that's why I was saying you need to be really diligent about scrubbing your equipment, and then also using a sanitizer like peracetic acid or chlorine dioxide to actually inhibit the growth of the biofilms, and they actually will remove them. The unfortunate thing is biofilms are resistant to many chemical cleaners and sanitizers. So there's a few like peracetic acid and chlorine dioxide, which are good at it, but many of them are not. Another unfortunate thing about biofilms, they can be baked on surfaces with hot water or steam. So in your bottling line, could be a problem if you're not properly cleaning the interior piping and you're having hot water or steam go through that piping, you can bake on the biofilms. They usually require physical action to remove from the surface, scrubbing, brushing, high pressure. And I know many wine spoilage microbes can form or join biofilms. Once they start growing in a biofilm, they can be sloughed off during production, contaminate wine, and accelerate spoilage. So I see biofilms in bottling lines often. So I get calls about people having issues with wine spoilage yeast in their finished product, and it's re-fermenting. And so I'll go out to the winery and inspect the bottling line. And what I find is that you get biofilms on the interior of the piping because they haven't been cleaned properly. And so when I'm swabbing those areas, you can tell that they haven't been properly cleaned and the biofilm is developed. And so basically what they're doing when this is actually after the filter. So after the membrane filters, you have biofilm inside the piping. The wine's passing across that biofilm and it's being contaminated every time it passes the biofilm. So this is why it's very important to make sure you're cleaning effectively on the interior of your piping and on the interior of your other equipment. So what are the common ineffective sanitation practices that I see at wineries? Lack of basic wine microbiology and sanitation knowledge. Ineffective cleaning prior to application of the sanitizer. This is what I was saying earlier. Cleaning is the most important step in sanitation. Ineffective or no sanitizer used following the cleaning process. So they've done a good job cleaning, but no sanitizer is used, or it's an ineffective one. Incorrect concentration for cleaners and sanitizers. It's very important to have the right concentration for the cleaners and sanitizers. Otherwise, they're not going to be effective. Insufficient time and temperature for cleaners and sanitizers. Frequency of cleaning and sanitizing. They're not doing it frequently enough, and so they're getting a buildup of organic debris on surfaces. Sanitation practices for oak barrels. This is one of the areas that I see a lot of issues, is the oak barrels aren't receiving proper sanitation. And as we know, Brettanomyces loves oak barrels. And so if you're not properly sanitizing or doing sanitation on the oak barrels, you're asking to get contamination from Brettanomyces, because Brettanomyces can go like 8 millimeters into the wood and survive in there. So it's very important to make sure the oak barrels are receiving proper sanitation. Sanitation practices for infrastructure, drains, floors, walls. You really need to make sure you're doing effective sanitation in those areas, because as I was saying, when microbes get on the drains and on the floors and they're just sitting there, they eventually get up in the atmosphere. The winery water system is not considered. Again, this is important because you don't want microbes in your water that you're spraying onto equipment after you've already cleaned it. So it's very important to have water be clean, and I usually recommend you have a filtration system for your water down to 0.2 microns. Awareness about the most problematic sanitation areas in a winery. So, wineries don't always understand that the drains are critical and the floors are critical, and the piping is critical, and valves on tanks are critical. So you need to be aware of what are the areas in the winery where I'm probably going to have issues with ineffective sanitation. Another thing is no periodic deep cleaning and sanitizing. So by this, I mean taking equipment, piping apart and actually cleaning it on the inside. This is very important. Somebody have a question? Okay. Lack of a schedule for non-daily sanitation activities. So this is called a master sanitation schedule. So those things which you don't do daily, like cleaning tanks and hoses and pumps, but like, you know, actually like infrastructure for surfaces, you should have a schedule on how often you do that. Sanitation SOPs. Even if wineries have sanitation SOPs, they don't always, their practices are inconsistent. So you need to have those SOPs laid out exactly what you're doing each time, and that you can train to those SOPs so that you don't have inconsistent practices. Little to no verification of effectiveness of sanitation practices. So there's no visual inspections, there's no ATP testing, there's no microbial swabbing. It's just hoping that whatever you're doing is effective. And so you need to have ways to monitor how effective your sanitation is. Julie had asked me to kind of put together a summary of the plating results for the 2023 uncorked samples from the Livermore wineries. So I plated a bunch of your bottle sterility samples, or bottle samples in 2023. And these are I just like to go over the results. And the results are indicating to me that sanitation practices need to improve because you're getting a lot of microbes inside in your finished bottles. So I plated a total of 154 samples. 37 of those samples had wine spoilage microbes. So 24% of the bottles had wine spoilage microbes. This included, and then the total plate of wine spoilage microbes themselves was 32. So 37 had microbes, 32 had wine spoilage microbes. And this included Acetobacter. There was no Lactobacillus, no Enococcus. There was a Pediococcus in one of them. Brettanomyces showed up 5 times, Saccharomyces 9 times, and then I didn't see any Zytosaccharomyces. So the fact, whenever I see Brettanomyces or Acetobacter in a finished product, that's indicating to me that probably the sanitation needs to improve. I don't see Brettanomyces in finished product very much at all. I've probably seen it 10 times since 2000. So the fact that you're seeing 5 of those have it is a little alarming. You should not be seeing the Brettanomyces in your finished products. I don't know how many of these wines were filtered versus unfiltered, but again, even if it's unfiltered, you shouldn't see this level of Brettanomyces in your finished product. Do you know?
Bob, is there, are these results available for the individual bottles that were sampled?
Julie, I would assume so, right?
Yes, I have the results.
Per,
Each individual wine, yes.
is there a chance I could get them for Cooter
Sure.
Ridge?
Yeah.
Thanks.
Uh-huh. And if I don't send it, just remind me.
I might get mine too, Julie, for Rosie.
Okay, so just to clarify, these wine samples came from the uncorked submissions from last year. So anyone that submitted wines to uncorked, they got a full chem panel, tannin analysis, and microbial analysis done.
Yes.
Yep.
Bob, does Velcorin kill Enococcus?
It can, but some of the species of some of the bacteria aren't killed fully by Velcorin. And Enococcus, I think, is at 200 parts per million. But like there's some species of Acetobacter or Pediococcus which are not, you have to actually go to 300 or 400. which is above the legal limit.
So because there's 0 Enococcus here, would you assume that everything was either filtered or Velcrined that you plated?
Not necessarily, because sometimes the Enococcus actually, after it does its job with malolactic fermentation, it tends to go into a viable but non-culturable state pretty easily. So once you have SO2 there, it doesn't really grow anymore. And these viable but non-culturable cells don't really show up in plating.
Okay, but those would show up in a scorpion.
Yes, I'm sure they would show up in scorpion, yeah. Yeah. But plating, not necessarily. Any other questions about this table?
Thanks for sharing that. I think it's good for people to understand why we think cleaning and sanitation is so important.
Yeah, and I agree, Julie. And the fact that there's quite a bit of Acetobacter and Brettanomyces tells me that there's things could improve, yeah. Okay, so let's talk about actual sanitation audit findings that I have during when I'm doing these audits. So I've been conducting sanitation audits since 2007. So I would say about 500 audits so far. And this includes 3 types of audits: baseline or survey audits, where I'm just trying to get an idea of how effective sanitation is at the facility. And then there's monitoring or follow-up audits. So I've already done an audit, and then I go back a year or 2 later just to see how effective they've implemented the sanitation practices. And then there's troubleshooting audits, where I actually need to go to a winery to figure out where the contamination is coming from. And I do a lot of these for bottling lines because you get stuff in product and then starts re-fermenting. So they need to know where the yeast, for instance, is coming from. So I'll actually go out to a bottling line and swab it and have the winery take apart piping and everything, and I'll swab inside everything. And so that's a troubleshooting audit. So I've audited wineries in Napa County, Sonoma County, Mendocino County, Central Coast, Central Valley, Sierra Foothills, Oregon, Washington, Ontario, Canada. And this year I actually went to Illinois and Virginia too. So I've been doing it all, kind of all over the country, mainly on the West Coast though. The wineries range from, small 1,000 cases all the way up to the very biggest wineries. And what I do is I audit, not always, but this is the kind of the scope of what I audit: the crush equipment, cellar equipment, bottling equipment, oak cooperage, infrastructure, atmosphere, and water. And then when I do, just one thing to point out about the baseline survey audits, I usually perform these on both cleaned and uncleaned winery equipment. Cleaned because I want to see how effective the sanitation practices are, and then uncleaned equipment because I want to see kind of a baseline: what does the equipment normally look like, while it's just sitting there. So these are the results for cellar equipment. And so what this graph is showing you, this is for baseline or initial audits. This is showing the average microbial load on various equipment. And so on the y-axis shows the average microbial load, and this includes yeast, bacteria, and molds. And then on the x-axis, it shows you the various equipment and what the average microbial load is I'm seeing during baseline audits. So the areas that show the lowest levels of contamination usually are tank walls and floors. The highest are tank rack valves and bottom valves, and wine hoses and wine lines. So when I do audits, usually the highest levels of contamination I'll see are for rack or for valves on tanks and for wine lines and hoses. This slide shows the results for, again, initial or baseline audits for bottling equipment. And the lowest, usually average microbial load, is for a filler bowl. which makes sense because that's probably getting the most attention as far as sanitation. However, the inlet outlet lines post the filtration system and the sparger rinser spouts show the highest levels of microbial contamination. So the problem with the inlet outlet lines coming after the wine filtration system is that you've already filtered your wine, and now you're contaminating your product because those are contaminated. The interior of the piping is contaminated. So a lot of times on body lines, I see contamination in the wine feed line post filtration into the filler bowl. So that line will have contamination, and so you're just contaminating your product as it's going to the filler bowl because there's nothing to protect it. The other one is the sparger rinser spouts. And this is problematic because if they're contaminated, you're either blowing contaminated air or contaminated nitrogen or contaminated water into your bottles. For the winery environment, the highest levels of contamination, which makes sense, are the floors and the drains. And then this table shows the highest frequency of wine microbes. So for crush equipment, the highest frequency of wine microbes is on presses, on conveyor belts, in sumps and pans. For cellar equipment, it's like the fittings, tank rack valves, and oak cooperage. For bottling, it's, like I said, the filter inlet outlet lines, the sparger rinser spouts, and the filter housings. And then for the environment, it's the drains and the floors. Those are the areas where I see the highest frequency of wine spoilage microbes. If we're talking about specifically about Brettanomyces, these are the 5 areas where I see the most contamination. Now I have to say that when I do the audits, finding Brettanomyces is not easy because Brettanomyces is a slow-growing yeast and it's outcompeted by other yeast easily. So you have to have a pretty high population of Brettanomyces on the surface for me to detect it. But when I do detect it, the highest frequencies are for rack valves, for fittings, for, again, hoses and lines, oak cooperage, primarily oak barrels, and then drains. I find it in drains. So that's why I was saying drains are important to make sure you're cleaning those. If we're talking about Zygosaccharomyces, another yeast, it's actually the same areas: tank, rack valves, small parts, fittings, wine hose lines, oak cooperage, and drains. And same thing with Acetobacter, the bacteria, again, valves, fittings, wine hoses, lines, oak cooperage, drains. So you can see a theme here. This is Lactobacillus, again, very similar. You can see a theme there. There's certain areas and wineries where I see a lot of these wine spoilage microbes. So this is the, I have one more section, Julie, is probably taking like 5 minutes. Was that okay?
Take your time, whatever you need.
Okay. So this is the last section. And what I'm trying to get across here is a correlation between ineffective sanitation practices and poor wine quality. So a winery has not done very effective sanitation practices, and this has resulted in the quality of the wine being poor. So this is the first case study. These are actual wineries. So this is where they had ineffective cellar sanitation practices. They had high levels of Brettanomyces and 4EP, 4EG in their cellared wines. And then when I went to the audit there, they had high levels of Brettanomyces on 2 pieces of cellar equipment, on a rack valve and on the interior of a wine hose. So this winery had Brettanomyces on their equipment, and it showed up in their wines. Another case study for ineffective cellar sanitation practices. This winery had a large VA spike in their cellared wines. When I went and did the audit at the facility, they had Acetobacter, Lactobacillus, Pichia, Candida, on the tank walls and rack valves. So if you recall, all of these yeast and bacteria can cause acetic acid formation. And the tanks at this winery were loaded with these wine spoilage microbes. So it's not a shocker that they had a large VA spike in their wines. Another case study was ineffective sanitation practices for oak barrels. This winery had high levels of Brettanomyces and 4E-G in their cellared wines. When I audited their barrels, this is actually 2 different wineries. They had high levels of Brettanomyces in the wood from the barrels. So you can see, like the one on the left, each sample I took had a high level of Brettanomyces. And then on the one on the right, they also had a high level of Brettanomyces inside their barrels.
Hey, Bob, this is Larry again from Cooter Ridge.
Uh-huh.
What's your recommendation on best practices for sanitizing barrels? So I know steaming is most widely used, but is there other ways to sanitize barrels if you don't have a steamer?
So I just should say that steaming is by far the best way you can do this. If you could all get a steamer, because steam actually penetrates into the wood, and you want to be able to steam for at least 5 minutes to really penetrate into the wood. If you're not doing that, you try to get the hot water as hot as you can when you're rinsing out the barrels. Other than that, you have to use chemicals, which is not great, right? You don't want to do that if you can help it. So is there a possibility you can get a steamer, or is it not possible?
Well, anything's possible, but
Yeah.
what about hot water and sulfur?
So could you put a small amount of like 180-degree water into a barrel with some sulfur in it and then close the bung and let it sit for a period of time? Would that help?
It could help. I would worry that the SO2 would volatilize pretty quickly. So I would say that the best thing you can do is probably, if you're not using a steam, is hot water, or, use a gamma jet or pressure washer with hot water for about 5 minutes. And then after that, you let the barrel cool down and use ozone. Do you have access to ozone?
I do not know.
Oh, okay. Because that's the other thing that you could try is at least hot water with ozone. That would be, because you'd be cleaning it out and then applying.
So as far as making sure when you empty barrels that you don't let them sit dirty for a period of time,
Yes.
and you get them rinsed, and make sure you visually don't see any residue inside the barrels. I think that's a really good start.
Yes. Yeah.
And then doing, when they're empty over the summer, for example, you do regular gassing of your barrels with sulfur. I think that's also really important. So some people will actually wash and sulfur their barrels on some kind of regular schedule. I think that's important when they're empty as well.
Yes, absolutely. Yeah. So it's just not, it's cleaning them as soon as you can after they're emptied. And then if they're staying empty, to maintain the sulfur inside of them consistently. But I do have to say that if people can get a steamer, that is the best way to do this, because it really prevents Brettanomyces, especially. Okay, so those were cellar examples. These are bottling line examples. So this winery had Zygosaccharomyces, Saccharomyces, and Acetobacter in their bottled products. The wines were filtered through 0.45 micron during bottling, so they were doing the membrane filtration, getting onto 0.45, which is good. They were doing a daily hot water sanitation at startup, a daily water flush at shutdown. And then when I did the audit of the facility, I found these microbes on their bottling equipment. So Saccharomyces was on the sparger, their filler spout had Acetobacter, and their leveler had Saccharomyces. So they were not doing an effective job on their sanitation. They were just doing hot water sanitation at startup, but no chemical cleaning at shutdown, just a water flush. And so that wasn't good enough. For this winery, they had Saccharomyces and Saccharomyces in their bottled products. They ended up having secondary fermentation in their bottled products. Again, the wines were filtered through a 0.45 micron during bottling. So they were doing proper filtration to remove all wine spoilage microbes. They were doing a hot water sanitation at startup. And then when I did the audit, they had the Saccharomyces and Saccharomyces at high levels on their bottling equipment post-filtration. So they were doing a great job filtering, but then the equipment post-filtration was heavily contaminated. So they were basically filtering the wine, and then it was being contaminated as it was going to the filler bowl. Another bottling line example, this winery had Saccharomyces in their bottled products. Again, they were having secondary fermentation going on in the bottled products. The wines were filtered through a 0.45 micron. They were doing daily hot water sanitation at startup, but the water was below 180 degrees, as I later found out. And then I found Saccharomyces all over their bottling equipment post-filtration. So again, they were inoculating their product, finished product, post-filtration. Okay, so that ends this session.
I think you scared everybody very nicely.
Okay, I hope it was nice. Yeah, so it is kind of scary because you need to protect your product, right? The only way you can really do that is have effective sanitation practices.
Hey, Bob,
Where
I
do
have
you-
a question. Sorry.
We'll do Griffin first.
Pertaining, thanks, Julie. Pertaining to walls and minimizing any kind of spoilage from microbes on walls, I've heard of a cheese producer who had a galvanized steel walls because the zinc in the galvanized was antimicrobial.
Yes.
And apart from the aesthetics of that, what are your thoughts there?
Yeah, I've heard of the similar things. And I know a couple of wineries that have done that sort of thing, and it works quite well. But it's expensive, right, to do that. Sure. And it's probably easier to clean.
It's easier to clean, you said?
Yeah. Because it's more of a smooth surface.
Alec, you had a question?
I'm good.
Okay.
I have a question.
Okay.
Bob, how, in terms of like ozoning the winery with gas, and first of all, before I ask this question, I understand that I need to monitor the amount of gas that's coming out in the time, but, let's just say you find TCA in your winery or other things like that. Can you use your ozone gas to kill that in your, barrel room?
Yes, it actually ozone will hydrolyze the TCA chemical molecule. So that is a way you could do that. Absolutely. Some wineries actually do the ozone gas in their cellars, like 2 or 3 times a week. They turn it on at night, and then when they come back in the morning, the ozone is dissipated. But the wineries the smell inside the cellars are much better when you use ozone like that.
Do you have an idea of like re-entry time on that? Is there some safety issues with your staff?
Yes, so you have to be very careful about that. So usually people, when they use it, they'll turn it on for like 2 hours in the middle of the night. And then there's actually a monitor you can use to see if it's dissipated when you arrive in the morning.
You could definitely tell, especially if it hasn't you know, if it's really, when you open the door, you'll smell it. So yeah, it's so strong. So
Yes, yes,
that doesn't mean that it's all still dissipated. So yeah, using the monitor is a really good idea.
yes. You have to use a monitor. It's very important for safety.
Can I ask you another question?
Sure.
So when I'm making a cleaner, and I've never ever really gotten a straightforward answer on this, but like, let's just say I'm going to make a 5-gallon bucket of hot proxy.
Mhm.
What is this standard, recipe, so to speak? So if I'm going to use 4 gallons of hot water, how much proxy do I put in per 4 gallons of hot water so that I can stir it up and know that that it's cleaning properly?
So for a proxy carb, the range of concentration is, I believe it's 7 to 14 grams per gallon.
Okay, that's pretty much what I'm putting it in.
Yeah, is
So-
that what you're using?
Is there a time limit on when you make something and how long you can use it and have it be effective?
Yeah, so as for cleaners, I always recommend they be made up in warm or hot water, because that's when they're going to be most effective. So as the water cools down, it's going to be less effective. So I think that's what you're driving at, right, Julie? Is
Yeah, so I'm
Oh,
just saying if you can't make it today and use it all week. You would want
Yeah.
to make it in smaller batches
Yeah.
and make it more regularly to get the full effect.
Yes.
I usually make I make a batch in the morning, and then before I clean everything to go home, I'll make another batch for cleaning at night or in the afternoon, evening, just if it's cold. But do you also recommend, the other thing that I use for sanitation, for rinsing things like valves and stuff, is like citric acid with a little bit of SO2 in it? And do you have a recommended amount for 4 gallons of water? And I was also told not to use hot water for citric acid.
Yeah, I wouldn't use hot water. You can use warm water though. It'll dissolve better, but not hot water. Yeah. I do, but I'll have to look that up, what that concentration would be.
Okay. Because
Yeah.
I've been using about the same as the proxy carb, but sometimes I feel like, I don't know if I'm not using enough, because sometimes it feels like the proxy carb, can be really slimy.
Mhm.
And I feel like I don't get all that slimy film off if I'm not, I feel like I have to add a little bit more citric in order to get it to the point where
You get rid of
I can get it off. So then I'll just, most of the stuff I'll ozone as well, but like for smaller things.
Yeah. Yes. Yeah. Yep. Makes sense. Have you ever tried alcohol to spray your valves and stuff?
I do use alcohol to spray my valves and everything, but I still clean them like after I do a racking and stuff. I'll still put them in the hot proxy and then put them in the citric SO2 before I put them away.
Yeah, makes sense. Yep. Okay. Yeah. So you want to basically know what the citric concentration should be.
If I have a valve on a tank and there's wine in the tank, then that's mostly when I use alcohol.
Got it. Okay. Yep.
So if you get that info, I can send that to Rosie for you, Bob.
Okay. Yes, that would be great.
Actually, that's one that, Julie, if you could send it to all of us, that's some
Yeah,
pretty
no
bad.
problem. Mhm.
So concentration for citric. Okay.
And if you use citric and you put, like, usually I'll use citric and I'll put about like 50 parts of SO2 in it, which is a really small amount. Is it necessary even to put the SO2? I mean, because SO2 is I've been told is a good sanitizing agent as well, but I wasn't sure.
Isn't it risky having them in the same solution together?
It is if it creates, well, it'll create like a gas,
Yes.
it'll offlet a gas. So that was why I was asking what concentration, because if you just put a quarter of a teaspoon of SO2 in with the citric, you won't create that. You won't have that at 4 gallons. Okay. You won't have that gas come off.
Right. But then how effective is that as far
Right.
as sanitizer? That's the thing.
That's what I'm asking, is if it's.
So is it better to just have a separate bucket of sulfur solution rather than putting them in the same bucket?
Yeah, probably. Yes.
And if you're going to use the sulfur solution, the SO2 mixed with water, do you even need to rinse with citric? Because I was under the impression that that like neutralizes the sodium percarbonate.
Yeah, that's what the citric is for. Yes.
Okay.
Yes. Yes. Are you opposed to trying peracetic acid for your sanitizer?
No, but I know, I understand it's really expensive.
It is expensive. Yes, you're right.
Yeah. If I could get away with not getting that, that would be good. But also I have an ozone machine, so
Okay. Okay.
except that you have to be applying the ozone water with the machine on, so you can't essentially fill up a bucket of ozone water and use it, you know, for a certain amount of time, because it has to be like hitting a specific surface for a certain
Right.
period of time in order for it. So the ozone isn't really effective unless I'm using it like on to clean a tank or spray on a bucket or something like that.
Right. That's absolutely the truth. Yes. Yes. Because it breaks down so quickly. Yeah.
I think one of the other things that you used to talk a lot about was cleaners or sanitizers where they have some kind of surfactant, so they stick to the stainless for a longer period of time.
Uh-huh.
Are you still recommending that sort of thing?
Yeah. So that gets back to the properties I was talking about on cleaners.
Mhm.
Where you should have those properties associated with your cleaner, not just straight chemicals. But should have those like surfactants and wetting agents.
So as far as like a residence time, if you wanted to wash a stainless tank, you would prefer that it doesn't like immediately start dripping down to the drain, but it
Right.
actually sticks on the stainless for a period of time.
Yeah, at least, some, minutes versus seconds or something like that. Yeah.
Right.
Yeah. And that's where the foaming product works well, like a foam cleaner, because that's sticking on the surface for an extended period of time and doing its job.
So Bob, it's Larry again. I wanted to get your input on one of our practices of kind of cleaning and sanitation. So like when we're done for the day, we'll clean the equipment or the hoses and valves and such with proxycarb water and citric. But we don't sanitize. And then when we go to use the equipment the next time, then we'll run it through the cleaner and sanitation using PAA. Is that, I think I know what you're going to say here, but is that an okay method?
It's not terrible, I would say. It's actually okay, but it's better if you actually do the cleaning and sanitizing when you're done. Because that
And then again, and then a second time when we're getting ready to use it.
Well, no, the thing is, if you've done a good job cleaning and sanitizing after you use it, and then when you go back, you should only have to sanitize it, right? Because you've already cleaned it.
Okay.
So,
Assuming it doesn't pick up any microbes as it's sitting.
yeah. So there's usually like a window of time, like I usually say 3 to 14 days. If it's just been sitting there, sealed up like a tank, for instance. Usually after those 3 to 14 days, you can just go back and sanitize it, and it'll be fine.
There's a guy who came to my winery from Gallo, and that's all he did there was clean and sanitize tank after tank after tank. And he said that they're allowed to leave them for 3 days and then they have to be recleaned.
Yes. Yep. I found that 3 to 10 or 14 days seems to be okay though.
Good.
Yeah.
So Bob did work at Gallo as well, so.
Oh, good.
He does know exactly what they do.
That must be based on your advice.
Yeah.
He gave them a lot of advice when we worked there. Yes.
Yes. So yes, if you can, because if you're doing a good job cleaning and sanitizing after you're done, then the surface is clean, right? And so you don't really have to really go back and do a thorough cleaning and sanitizing again when you go back to use it.
Okay, I guess the thing would be to do it both times, the full cycle both times.
Yeah, no, you shouldn't have to.
More never hurts.
No, more never hurts, but again, you're wasting your water, time. So if you
money,
money, yep.
So I know some people will just do a water rinse when they are going to go back in. So they're not doing the sanitation. They've done the cleaning and sanitation when they empty the vessel.
Uh-huh.
But when they go to do work the next time, they're doing a water rinse. How do you feel about that?
No, they should use a sanitizer. Yeah. At least a sanitizer.
And if you know that your city um, water uses some kind of um, bleach in the city's water system,
Mhm.
how do you feel about that? Do you feel like you need to set up some kind of water filtration system coming into your winery?
Yes. You need to use an activated carbon filter. Yes, because that chlorine is coming into your facility and it's sitting in there at that point.
So your potential for TCA on your entire facility
Yes.
is pretty high just from using water from the city.
Yeah.
So
I
if
would
you're
say it's pretty high. I would say there is a risk. Yes.
okay, so if you're saying somebody that feels concerned about the water in the city of Livermore, and so they, whenever they make water additions, they feel they should purchase I don't know, carboys of water to use,
Mhm.
then they already know there's a risk in the water in the winery. So
Yes.
at that point, you should set up some kind of water filtration system just to protect your winery from any issues you might have.
Yes. Yes. And in particular for TCA and for chlorine or bromine, you want to use what's called an activated carbon filter, because that'll actually remove those compounds, chemical compounds from the water.
And Bob, if a winery is on well water, you recommended filtrating down to a 0.2 micron filter.
Yes.
Is that right? And
Yes.
anything else beyond that?
I would have a pre-filter like a 1 micron and then a final filter 0.2 micron. So that, you know, the pre-filter protects the 0.2 micron filter.
Then probably doing regular monitoring to make sure
Yes.
the filters are working properly.
Yes. Integrity testing. Yeah.
Mhm.
Yeah. Yeah, because there's microbes that are smaller than 0.45 out there. And for the wine industry, 0.45 takes out all the wine spoilage microbes. But there are microbes that can get through a 0.45, so that's why I go down to a 0.2.
Anybody else have any questions for Bob? So if you have other questions that you think of between now and our next and third part in the series, you're welcome to ask those questions too. You don't have to ask them all here in front of everybody. Thank you, Bob. Did you want to just say a little bit about what our next meeting will be about?
Yes, so next time I'm going to talk about, first of all, the benefits of a sanitation program. So, and I'll provide examples of why this is important. And then I'll be actually going through my recommended sanitation practices for harvest equipment, for cellar equipment, for plowing equipment, for infrastructure, for your water system. So some of the questions that you were asking today about what do I use for this situation and how much, I'll be going over some of that stuff next time. And then the final part of the next session will be talking about the impact of improved sanitation practices on reducing microbial loads at the winery. So wineries have had me come in, I've done an audit, I then have recommend some sanitation practices, they implement them, and then when I go back, what do the wineries look like after they've implemented it? And you'll be able to see a pretty dramatic difference in their microbial loads on equipment and infrastructure services after they've implemented these improved sanitation practices. So that's generally what I'll be talking about next time.
Thank you. He does offer sanitation audits. He's kind of busy with that, but he does offer that, and he can, if you give him some information about the size of your winery and how much equipment you have, he can give you a quote on how much it would be to get an audit from Bob. Just to give you an idea, there are some folks in Livermore that have taken advantage of that already. But it is an option for you guys if you really want to know how your winery looks. He does do that sort of thing. So you can ask me to give you his contact info, or you can copy him. If you have his email already, or I can get it to you.
Yeah, sounds good, Julie. Thank you.
Mhm. And
Julie, can I ask one more question? Just before we
absolutely.
close?
Yeah.
Bob, how much contact time do you recommend for cleaning up like hoses?
Usually like, I'd say 10 to 15 minutes, usually pretty good.
Okay, thank you.
Mhm.
Well, thank you, everybody, for showing up, and thank you, Bob, for all this information. I think
course.
people got a lot of insight out of it, so really appreciate your time. And anybody, if you have questions, we're always available for answering them. So
Yes.
thanks a lot, Bob, and we will see you in July.
Thank you, Bob.
Thanks for everybody coming.
Thanks, Bob.
Right. Have a great
Bye.
day.